Welcome for this special edition of, I think we should call this the Don and the Dr. Don Layman and Dr. Gabrielle Lyon show. We'll just
Something like that.
something like that. Uh, Don and I wanted to jump on here. This might take longer than
Friday chats. Let's just go with Friday chats.
this will be our forever strong Friday.
There you go. I like that.
This will be okay, great. So we've agreed on the Forever Strong Friday. And today we are talking about the paper that has absolutely been the center of conversation from Cell Reports Medicine. And that article is we'll post it here so you can see what it is. The anabolic response to protein ingestion during recovery from exercise has no upper limit in magnitude and duration in vivo in humans. The lead author is Jorn Tromlin. I may be saying that wrong. Tromlin. This is out of Luc van Loon's lab. All right, let's kick it off. Dr. Layman, let's talk about the smiling. Let's talk about the title in and of itself. The anabolic response to protein.
I think one needs to start by saying this is a really good group. In my mind, this may be the best group in the world in doing protein metabolism work right now in terms of looking at different kinds of proteins and looking at plant versus animal proteins and looking at older adult, younger adult, just sort of the whole package. This may be the most active and best group. So, as we wander into sort of looking at this paper, everybody needs to at least have the context that these guys are really good. And this paper seems a little out of character for sort of how they bracketed their conclusions. And we can start with the title. I think the title suggests things that aren't really true.
Let's talk about that. The anabolic response to protein ingestion.
Let's stop there. Protein ingestion. So that kind of implies any protein you eat is all equal. And what we know from their work is probably the single most unusual and slowest protein to digest and absorb is casein. And this study is done with casein. So that makes it odd to start with. They use milk protein for their treatments, and milk protein is 80% casein. So first of all, start out, it's not protein ingestion, it's casein ingestion. Okay, go on.
Well, wait, before I have a few clarifications that I think are important. Could you explain when you say casein versus a whole protein? What do we know about casein? What do we know about the time of absorption, the impact? What do we know that makes casein different?
So there's a lot of work with isolated proteins, whey proteins and soy proteins, pea proteins, wheat, gluten, etc., and quite a lot with casein. And what we know among all those isolated proteins is casein probably digests and absorb about 4 times slower than any other protein. And it's because it's the protein of cheese. And so when it gets in the acid environment of the stomach, it curdles. And so it's very slow to unwind and get digested as it moves into the intestinal tract. So we need to keep in mind that they selected a very unusual protein for this.
And I think that that's really important.
And I mean, the reason they selected it is they have this very unusual model where we use tracer technology to monitor a lot of these things. And what that means is in the old days, we used to use radioactivity, but now we have what are called stable isotopes. And basically they fed these isotopes to a cow, and the cow produced milk. And then they isolated the proteins from the milk. So they have what we call intrinsically labeled proteins. The label's actually in the food versus usually when we do experiments, we use a tracer that's separate. So this is a really cool study, but unfortunately, you have to keep in mind they're using an unusual protein.
Yeah. And they did not specify that. So the anabolic response to protein ingestion during recovery from exercise.
So we can stop there again.
Okay.
Recovery from exercise. That implies that any exercise you and I would go out and do in the morning is going to have this response. But that's not really true. What they did was they took untrained young adult males, and what they did was an acute exhaustive resistance exercise. They exercised them to failure with 10 different exercises, 10 different failures. So this is an absolutely extreme one-time kind of effect. It's not normal.
Right. And they say that it has no upper limit in magnitude and duration.
So the anabolic response, no upper limit in magnitude. That means you can basically add protein and gain muscle forever. And we all know that's not true. Just eating more protein, eating a bigger steak doesn't give you bigger muscles. So just the suggestion of that seems so off base. The duration issue makes some sense for what I already mentioned, is that they use casein. Casein is extremely slow to digest, and so it extends the duration. So that kind of makes some sense, and we can kind of look at what that means. And the last line is in humans, which means all humans get the same effect. But it's important to recognize they used young males under probably the age of 30, but they actually don't even specify that in the data. So we don't really know. But basically, it's certainly not all humans.
Right. And I
So
also,
other than that, I have no problem with the type.
that is hilarious. And I just, before we kind of dive into this, I want to ask you something, and then we'll take a look at the summary. Maybe we'll actually, instead of to the summary, perhaps we skip and we look at their results and their hypotheses. I'll let you decide. But my question that I'm going to pose to you is Cell Reports in Medicine. Is Cell Reports in Medicine a nutrition journal?
No, it's really more of a molecular biology journal. It really likes this kind of a study, which has a lot of molecular data, genetic data, metabolomics data, is the type they want. I've actually criticized this journal some in the past for lack of clarity in nutrition designs. And I think this one sort of has that markings that if you look in the paper, you can't even find any of the methods. I mean, the methods are in supplemental tables that you have to go online to find. And even there, they don't really specify what the diet was. They say it was milk protein. So if you know milk protein's 80% casein, you can sort of begin to backtrack all of this, but it's not stated. It's not, the methods aren't clear. And like I said a minute ago, they don't even designate what the age of the subjects are. They say they had subjects between 18 and 40, but in previous papers where they've said that, it turns out most of the subjects are around 27, 28. So, that's, 28-year-olds are definitely different than 40-year-olds.
Right, right. You and I talk about that quite a bit. Where do you feel that would be the next place? My next discussion point would be what they chose. So we know that they make some mention about skeletal muscle being in this constant state of turnover. They discuss this stable isotope methodology. I do think that it's much more complex, that they do use more advanced, and it seems to be novel ways in which they are looking at these tracers. And then in this third paragraph here, they talk about a dose response, and they talk about how studies have reported that the ingestion of 20 to 25 grams of protein is sufficient to maximize post-exercise muscle protein synthesis rates in healthy young adults, with no further increase in muscle protein synthesis rates when larger amounts of protein are consumed. Let's stop here and talk about the dose that they chose, with the fact that the dosing was from casein.
Exactly. So they're correct in saying that there have been multiple research studies, I think some even in their lab, others in Stu Phillips, others with Bob Wolf, et cetera, where they've looked at uh, protein dosing after exercise. I mean, we did the original studies in animals where we showed that exhaustive exercise would decrease protein synthesis, and then we could look at recovery. People have looked at that in humans after resistance exercise, and what you find is in young males, you will typically plateau that with about 20 to 25 grams of whey protein. But what I already mentioned is that this is casein. And what the same group, Van Loon's group, the Netherlands group, has shown is that casein has less than half the impact. They've shown it in older adults, they've shown it in younger adults, is that typically to get casein to have an effect, they have to go up to more like 45 grams.
Yes.
And so now we get into a very interesting design problem. So they have used 20 grams of protein and compared it to 100 grams of protein. And so they're going to say, and now they're saying that there's no upper limit to the magnitude simply because 100 gave them higher results than 25. Well, the problem with that thinking is 25 grams of casein is too low.
Right.
And so what we know is that that number typically plateaus at 45, maybe 50. And so now what we need to really make the claims they're making is they need more of a titration study where they have a 50 value or even a 75. My guess is the values would actually plateau by 50, maybe 55, I don't know. But the difference between 55 and 100, I'm pretty sure wouldn't be significant. So the idea that it takes 14 and a half ounces of steak to reach a recovery level of protein is silly because we know it's not that high.
Yeah, it was, somewhat odd that they talk about protein, and everybody in the protein space knows that casein, it has these characteristics.
Yeah.
So reading this, looking at this dose response study, and I've seen this all over social media about how, wow, this 20 to 25 gram of protein was sufficient to cause this response. And we're all thinking, yeah, these guys are exhaustively exercised, they're super young, and they're using a protein source that nobody typically uses interchangeably with protein. They
Yeah.
call it casein.
I mean, the 25 gram response sort of does what you'd expect it to do. But the problem is they then go to 100 and imply that that's some sort of a maximum number. And my problem with it is based on their own literature. I think it would plateau at 45 or 50. So they chose 25. If they'd have actually made their first dose at
40.
45, they
Yeah.
would have seen no additional
Right.
difference between 45 and 100. So that's kind of a misleading technically I would call it a control group. They need a titration study to make the points they're making. And they didn't do that. They picked a low number, and so anything higher than the low number was going to be higher. And they happened to pick 100, but I think they could have picked 55 and they had exactly the same results.
Yeah. I obviously agree with you.
I mean, one of the other subsets of that is they got into digestion. And what they showed is that, and it gets back to the title and duration, what they showed is that after this 100 grams of casein at this morning breakfast meal, 12 hours later, they had only digestion absorbed 53% of it.
Yeah, let's pull
That
that out.
means that means 47 grams of it have
Right.
disappeared somewhere. Where did it go?
Yeah, and where is that? I do remember reading that, and I think that that's important.
It's late in the paper.
Mhm.
And again, it's based on this isotope work. And so then we have to begin to bring into question the isotopes. They're basically using multiple isotopes and trying to conclude how much of the protein showed up in the blood. What we know from multiple digestion studies and from PD-CAS and DIAS is that 12 hours after a meal, you don't have 50% of the protein still in the GI tract. So
Right.
that just doesn't make any sense. So the question is, where did it go? They've got 47 grams of protein unaccounted for after 12 hours.
So where do you think that it was, so, and I think that it's fair to say that these are more novel approaches, these stable isotopes that they're using. You had mentioned that they're using, um, they did you say three different isotopes?
They actually have 4 different isotopes in this study.
Okay. Which for the viewer or the listener, this is, that is much more robust. Typically it's one.
Yeah. Yeah. The majority of the studies I've ever done had 1, and I've done a few with 2. I've never even thought of doing one with 4. But again, I think it's cool. The research is cool. I think the study is a very interesting one, and it raises some very interesting questions. But the conclusions they've jumped to are really overextensions of the data.
Well, what about this idea that they're missing 40 some percent of the protein ingestion? Do you think that that's just an isotope error? It's not accounted for? How
I
would
I
that
don't know
make
what
you
I
guys?
don't know what to think of that. I mean, 100 grams of casein at a dose, I have nothing to draw on for that. I don't know how slow that would digest. It's hard to think about how to relate to that. I mean, that would be like um, that would be like eating 8 yogurts at one sitting.
Right.
I don't know, I just don't know how to think that through. But that's what their data says.
Mm.
So again, they're talking about there's no limit to the duration, but it's because they've somehow altered the absorption pattern uh, in ways that aren't normal.
Mm.
So again, I question the duration issue because I think it's totally unique to the protein they chose, casein.
Yeah.
So
Yeah.
if they'd have started the title that the anabolic response to casein ingestion after exhaustive endurance exercise extends the duration of the response, I'd get that. But that's not what they said.
And then, so where do you think is the next kind of highlighted
I
area
think
here?
I think the next thing that sort of stands out to me, and again, I don't know quite what to make of it, is that they say the meal doesn't matter, but yet what they did was the experiment after the overnight fast. So
Yes.
we all know that breakfast is the most sensitive. So if the meal pattern doesn't matter, why'd they choose breakfast? Why didn't they choose dinner? They have shown in a study just in 2023, the same group, the same author actually showed whey versus casein, 45 grams, that casein was far inferior. I'm sorry, yeah, casein was inferior to whey protein. In fact, casein, a 45-gram meal after dinner, made no effect on protein synthesis.
I mean, that's important. I'm seeing if I
I
can pull
mean, I
up.
mean, so the same authors have conflicting data on that. So now they've chosen breakfast, which is far more sensitive, and they've now gotten groups, people, and then they did this extreme exercise. And if you look at their signaling data, mTOR, you and I talk about EIF4, BP1, the binding protein, S6, these are all the initiation factors. And what they found is that there was no change in these after the meal.
That can't possibly be correct.
There are dozens, probably hundreds of papers showing the changes in mTOR and EIF4 after a breakfast meal.
Right.
They suggest that it's a rapid change and that it becomes irrelevant. So their first measurement of that is, I believe, six hours later after the meal. I'd have to look at that again.
I'm
And
looking for it
so
right now.
and so they've measured that, somewhere out around 6 hours, and they're not changed. That doesn't make a lot of sense. However, if it's true, what that says is this: exhaustive acute resistance exercise is triggering muscle protein synthesis by mechanisms that are abnormal. And that may be true. That may be true, that this acute damaging resistance exercise treatment is altering the way that muscle regulates anabolic responses. They refer to it as a recovery response, and I think that's true. I think this is a one-off recovery model. That's interesting. If it's triggering it by a mechanism other than mTOR, that's really cool to explore, but it's totally inconsistent with any data in the literature.
Which I think is important to point out. It looks like they said through the initial 4-hour postprandial period.
Yeah. And so what we have shown and others have shown is that after a reasonable protein meal, more like a 30, 35 gram protein meal, that mTOR, EIF4, S6 ribosome are all still elevated at 5 hours. And at 4 hours, they show that it's not different than the fasted control. So that's totally different than what multiple groups have published.
And what I think is also interesting is it may be different than the group themselves. So they don't really make any mention of the multiple studies that they have done. Or previously, there's multiple Van Loon muscle protein synthesis and sleep, whey versus casein. Dose response of muscle protein synthesis after aerobic exercise, etc.
And we know that exercise lowers the threshold. So, if you were going to maximize your protein response, the things you would do is you'd pick the morning time after an overnight fast, you'd pick young males, and you'd pick resistance exercise. So they picked all three of them. So if they're going to maximize the effects of what they're looking at, they picked all three categories.
Yeah, it's truly fascinating.
Yeah. And they're arguing now that meal distribution doesn't matter. And I think there's some extent that's true, but I think it's particularly true in young males. I think that if you look at the literature about meal distribution, the literature says it's important in older adults, people over 40. This is a study of people under 40. And so in older individuals, particularly if they're under any catabolic stress, say they're sedentary or trying to lose weight, we think that first meal is critically important. We think having super large meal late in the day isn't very beneficial. And in fact, the same group, as I mentioned a minute ago, showed exactly that same thing in 2023. They showed that whey versus casein in a meal post-dinner had the casein had no effect at all. So we know that the first meal is probably the most important, and yet that's what they used, and then concluded the meal wasn't important. So if they really believed that, they should have done a dinner meal.
Yeah. And what was the ultimate takeaway, do you think, in terms of what did we learn from this study? What are the statements that are being made? And then what could we actually say from this study?
I mean, one of the statements that catches my eye is they say that, again, it has no upper limit and there's no oxidation of amino acids after these meals. That is totally inconsistent with everything else that's ever been published. We know from the indicator amino acid oxidation methods, we know from multiple other routes, we know that leucine specifically, when it increases in the muscle, it triggers the branched chain ketoacid dehydrogenase, the mechanism for oxidation. So we know that increases in intracellular amino acids trigger oxidation, and yet they failed to pick it up. So does that mean this model is that unusual, or that their tracers are insensitive to that? And I can't tell. I've never done these tracers, but we know that oxidation's got to follow. And if in fact it doesn't, that means that all of this protein is going directly into protein gain. And if you track that out, that would mean they're going to gain at least a pound of lean body mass every 12 hours. I mean, we just know there's no such gains in that kind of realm.
Mm.
And so, again, there's just a lot of things that don't extrapolate out. So, what did we actually learn? We learned that exhaustive, acute resistance exercise in untrained individuals has pretty dramatic effects. And we also relearned that casein is a very slow digesting protein. And so when you put those two together, we get a much more prolonged recovery response. I think that's all interesting, but it frankly doesn't really change any thinking. I think that we know that exercise, resistance exercise, enhances sensitivity to protein, that there is a recovery response. And we know that in trained individuals, it can be pretty prolonged. I mean, we can have a benefit of the exercise for 24, 48 hours. I think those are all positives we learned from it. And this acute exhaustive resistance exercise is kind of an interesting model that I don't know that anybody's really used. I don't know. I think it may be useful to sort out mechanisms, but frankly, it's not very practical.
Right.
You can only do an exhaustive resistance exercise once. The second time you do it, it's not acute. It's the
Yeah.
second time.
So interesting.
So anyway, I think it's fascinating by a really good group, complex mechanism, but they've kind of got out over the end of their skis here with their conclusions.
Yeah. Well, thank you, Dr. Lyman. This was so helpful for the listener, and we'll pair some of the other papers, their history, so you can learn and read more about it. And again, thank you so much.